And our study program, Magister Ilmu Laboratorium Klinis or Clinical Laboratory Science, and Research Center for Marine and Fisheries Product Processing and Biotechnology, Kementerian Kelautan dan Perikanan through Dewi Sesita Zilda, which now part of BRIN. There are many other important guests that are here, which I could not fully mention, from Magister of Fisheries and Marine Science, Universitas Diponegoro, Faculty of Biology and Marine Science, Universitas Hasanuddin, National Research and Innovation Agency of Indonesia, or BRIN, and also Asian Federation of Biotechnology, Research Center for Marine and Fisheries Product Processing and Biotechnology, Kementerian Kelautan dan Perikanan, and then Ikatan Alumni Teknologi Laboratorium Kesehatan Unimus, or Alumni Association of Medical Laboratory Technology. I hope you are all in a good health, and I hope you can enjoy this forum. So now let me announce the rundown program for this morning's event. First, we will be singing Indonesian National Anthem, Indonesia Raya, and then singing Hymne Muhammadiyah, and then welcoming speech by Rector of Universitas Muhammadiyah Semarang or the representative to open the forum. And then photo session. So as wished, audience could use Zoom background that we share in chat room. And then followed by opening speech from Professor Oki Karnarajasa, PhD. I hope for 20 minutes, Prof. So you can share any stories with us. Yes. And then the core of the event is lecture from Professor Russell Hill. As requested by Professor Russell Hill, will be up to 1 hour or less to allow discussions. From there, I think we will finish around 10 EM. Otherwise, very late for Professor Russell, but I hope it's okay for if more than 10 PM there, Prof. Russell. So I suggest students who want to raise questions, write down your questions in a short and a precise way so that we can read all the questions today. Not too long, okay? Because we have very limited time. And also don't forget to fill attendance form to obtain certificate of attendance from us. Ladies and gentlemen, let's now join opening ceremony by singing Indonesia Raya Song and Hymne Muhammadiyah. Please, Mbak Fitriani. The next will be singing. We will be singing this Muhammadiyah.
Tadi biar nggak nggak menyala ini mati turun dia.
Sorry, so please the mute the other. Yes, ladies and gentlemen, lecturers, researchers, and students, now it's time for welcome speech by Rector of Universitas Muhammadiyah Semarang or his representative. Rector of Universitas Muhammadiyah Semarang could not be here with us, but he will be represented by Vice Rector of Academic Affairs, Dr. Budi Santoso, MSI Med. So I welcome Dr. Budi Santoso to address welcome speech. Please.
Baik, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning everyone. Respectable Professor Russell Hill, PhD from Institute of Marine and Environment Technology, University of Maryland Center in USA. Yang kami hormati Profesor Oki Karnarajasa, PhD dari Badan Riset dan Inovasi Nasional, para mahasiswa S2 ILK Unimus, dan seluruh peserta kuliah umum pagi hari ini. Alhamdulillah, Prof. Russell Hill, Prof. Oki, dan seluruh tamu undangan. I would like to welcome warmly to Universitas Muhammadiyah Semarang, especially to Master of Clinical or Medical Laboratory Science at General Lecture, even though by virtual. The Master of Laboratory Science Study Program is one of 28 study program currently owned by Unimus. And the study program is the only one in Indonesia. The purpose of medical laboratory science education is to product scientists in the field of medical laboratories that work to analyze a variety of biological specimens. They are responsible for performing scientific testing on sample and reporting result to physician. The data they find plays an important role in identifying and treating cancer, heart disease, diabetes, and other medical conditions. Ladies and gentlemen, medical laboratory science collaboration very closely with physician and medical laboratory technician in diagnosing and monitoring disease process, as well as monitoring the effectiveness of therapy. Area of medical laboratory training includes microbiology, chemistry, hematology, immunology, transfusion medicine, and toxicology and molecular diagnostic. Bapak dan Ibu saudara sekalian, ingin saya sampaikan dalam kesempatan pagi hari ini kepada seluruh peserta general lecture, tentu kami sangat senang, sangat berbahagia pagi hari ini kita bisa sharing ilmu bersama-sama, dan program studi laboratorium medical laboratorium science Unimus ini adalah yang pertama satu-satunya di Indonesia sesuai dengan rekomendasi organisasi profesi dan juga dari Dirjen Dikti, bahwa diharapkan satu program studi ini bisa menghasilkan para saintis di bidang laboratorium klinik atau laboratorium medis yang memiliki kualifikasi kompetensi di bidang laboratorium, bisa menganalisis cairan dan jaringan tubuh manusia untuk keperluan diagnostik kesehatan. Dan tentunya di dalam kuliah umum ini diharapkan irisan dari ilmu pengetahuan yang lain yang sekiranya itu bisa dimanfaatkan, saya kira sangat prospek untuk meningkatkan kompetensi di dalam capaian anak didik kita di bidang sains laboratorium medis. I hope the general lecture will give benefit to all of us, and also I hope the general lecture will contribute their experience and expertise so as to improve our knowledge. Saya kira cukup, mudah-mudahan apa yang hari ini bisa kita dapatkan di dalam general lecture ini bisa meningkatkan kompetensi kita di bidang ilmu laboratorium klinik. Terima kasih kepada seluruh panitia yang telah menyelenggarakan kegiatan ini. Selamat pagi, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Budi. And ladies and gentlemen, now we want to show you the company profile of Magister of Clinical Laboratory Science. Please enjoy.
Sains Laboratorium Medis, Fakultas Ilmu Keperawatan dan Kesehatan, Universitas Muhammadiyah Semarang. Mempersembahkan profil program studi. Program studi Magister Sains Laboratorium Medis Universitas Muhammadiyah Semarang berlokasi di Kota Semarang yang merupakan ibu kota dari Provinsi Jawa Tengah dan merupakan salah satu kota besar di Indonesia. Program studi Magister Sains Laboratorium Medis diselenggarakan dalam rangka mendukung upaya peningkatan mutu pelayanan kesehatan di bidang laboratorium medis dan mengikuti kemajuan teknologi laboratorium medis yang berkembang dengan pesat. Program studi Magister Sains Laboratorium Medis merupakan jenjang pendidikan lanjut bidang vokasi program studi analis kesehatan, mulai jenjang SMK Analis Kesehatan, D3, dan D4 Analis Kesehatan. Saat ini di Indonesia belum ada program studi Magister Teknologi Laboratorium Medis. Program studi Magister Sains Laboratorium Medis Unimus menjadi program studi pertama kali ada dan satu-satunya program studi yang ada di Indonesia. Profil lulusan Magister Sains Laboratorium Medis terdiri dari peneliti, konselor bidang teknologi laboratorium medik, inovator, manajer laboratorium medik, tenaga pendidik, dan ahli teknologi laboratorium medik. Program Studi Magister Sains Laboratorium Medis menjalin kerjasama dengan berbagai instansi baik dalam dan luar negeri untuk meningkatkan proses pembelajaran kelas dan klinik serta meningkatkan kualitas dosen. Di antaranya Prodia, Proline, Prestem, LIPI, Balai Industri Biologi Laut, Kementerian Kelautan dan Perikanan, Balai Besar Riset Pengolahan Produk dan Bioteknologi Kelautan dan Perikanan, Laboratorium Fasilitas Penelitian Bersama Fakultas Biologi UGM, DPP dan DPW Patelki Jawa Tengah, Universitas Kanazawa Jepang, Saint Dominic College of Asia Filipina, Mahidol University Thailand.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Airul Arham, salah satu mahasiswa Magister Sains Laboratorium Medis. Saya di sini sedang melakukan penelitian PCR untuk amplifikasi 16ES. Saya bangga menjadi bagian dari mahasiswa Unimus karena di sini memiliki fasilitas laboratorium serta dosen yang berpengalaman dan berkompeten di bidangnya masing-masing. Saya di sini belum lulus tapi sudah diminta bergabung bersama Politeknik Muhammadiyah Makassar. Ayo bergabung bersama kami di program studi Magister Sains Laboratorium Medis Unimus.
Kegiatan mahasiswa program studi Magister Sains Laboratorium Medis diantaranya 1. Short course 2. Pembicara seminar dan workshop.
Perkenalkan, nama saya adalah Eleveti Oklarina Putri. Saya merupakan orang yang beragama non-muslim, tapi di tempat ini saya diterima dengan baik oleh teman-teman, oleh dosen, kemudian oleh orang-orang yang berada di lingkungan ini saya diterima dengan baik. Saya bangga menjadi mahasiswa Magister Sains Laboratorium Medik karena di sini didukung oleh fasilitas-fasilitas laboratorium yang lengkap, fasilitas-fasilitas kelas yang lengkap, serta didukung oleh dosen dan laboran-laboran yang kompeten di dalam bidangnya masing-masing. Ayo jangan ragu untuk bergabung bersama kami di program studi Magister Sains Laboratorium Medis Unimus.
Berbagai sarana dan prasarana lengkap disediakan oleh Unimus guna menunjang pembelajaran di program Magister Sains Laboratorium Medis, antara lain Laboratorium Biologi Molekuler, Laboratorium Mikrobiologi, Laboratorium Patologi Klinik, Laboratorium Hematologi, Laboratorium Research, dan Laboratorium Hewan Coba. Guna mendukung iklim pembelajaran yang kondusif, Universitas Muhammadiyah Semarang menyediakan berbagai fasilitas sebagai sarana untuk beribadah, berkumpul, berdiskusi, ataupun menggali ide tentang perkuliahan di Magister Sains Laboratorium Medis, seperti perpustakaan yang telah terakreditasi A, ruang CBT, gedung pusat kegiatan mahasiswa, gedung serbaguna, Masjid At-Taqwa Muhammadiyah, Jawa Tengah. Taman dan gazebo. Area parkir yang luas. Unimus Mart. Serta sport center seperti lapangan tenis, lapangan basket, lapangan futsal, panjat tebing, dan lainnya. Unimus, a university for the excellent.
Okay, so now is the next session is second welcome opening speech by Professor Oki Kana Rajasa. Let me read shortly his CV. Professor Oki Kana Rajasa is the Deputy Chairman of Earth Science at Indonesian Institute of Science or LIPI and was a former Director of Research and Community Service, Ministry of Research and Technology of Republic of Indonesia. We still remember in his period, grants were so generous with awards to most productive lecturers or researchers. So up to 4 projects at that time. So as principal investigators. He also initiated national postdoctoral grant, and it was a great experience, and I hope that that experience will come again, Prof. Prof. Oki was previously appointed in various positions in Universitas Diponegoro, including expert staff of Rector for Development and Cooperation, Head of the Integrated Laboratory, Head of the Community Research and Development, and Coordinator for Research and Development of Universitas Diponegoro in 2014 until 2019. Prof. Oki was member of the Research and Development Commission at the Ministry of Marine and Fisheries. Prof. Oki published 100 of high-impact scientific papers. He is a recipient of several awards, including Collaborative Development Award by British High Commission Singapore, British Council, Biovision Catalyzer Award on Biodiversity, Climate Change, and Agronomy at the Biovision of Wildlife Science Forum, France. An Equipment Award from the German Ministry of Economic International Cooperation, from Cipta Lestari Kehati Award from the Indonesian Biodiversity Foundation, Kehati, and 5-year Biology Award from the Faculty of Biology of Universitas Sudirman. As we know, Prof. Oki is the alumnus of Faculty of Biology, Jenderal Sudirman University in 1989. He completed MSc program in microbiology from Ontario, Canada, and obtained his PhD in marine microbiology at the Department of Aquatic Bioscience, University of Tokyo, Japan. I welcome Prof. Oki to present his opening speech.
Okay, thank you for the introduction. I would like to share some slide. Can you see my slide?
Bisa Pak Prof. Oki.
Okay, alhamdulillah.
Yes, bisa.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Russell, and very good morning, everybody. It is my great honor to be given opportunity to welcome Professor Russell Hill to give his general lecture. I put the title of my welcoming remark is that drugs from the sea, it is no more a myth. I think, Russell, you remember in 1978, Cole and Siderman published a book with the title of Drugs and Food from the Sea, Myth or Reality. So I think today's lecture is a journey progress on the drug from the sea that I believe Professor Russell will be given us and brief up on the new development of this study. Why this is important for Indonesia? Because Indonesia is part of the global center of marine abundance and diversity, known as the Coral Triangle area. And Indonesia occupies most of this global center for marine diversity. When we talk about the marine biodiversity, diversity means chemical diversity. So I think it is very important for us to be able to understand the phenomena that happen within our marine environment in order to better understand the potential of marine environment, especially those from the marine biota as well as from marine microbial community. One of the most important development that stimulate our understanding on the marine natural products is that chemical ecology and natural product chemistry has been the most important and significant development, especially to clarify the chemical basis of ecological and behavioral interaction in the nature. Our understanding on the marine chemical ecology contributes very significant on the development of marine natural product chemistry with different function and purposes, such as for the industry, medicine, and agriculture. Our understanding on the role of secondary metabolites, especially for the marine chemical ecology, also inspire us to work and explore more potential function of the secondary metabolite that exists in the nature. For example, for the microbial effects, this will allow us to develop our potential of the secondary metabolite, especially for the development of antimicrobial compound. The antifouling potential of secondary metabolite also has inspired us to develop natural antifoulant that is very important for the development of maritime industry. I'm very pleased to inform you that our works on health and maritime, especially to seek for the bioactive molecule from marine environment, has been within our national research priority between 2020 to 2024. And why we are very much concerning about the marine microbe, I think one of the reasons is that there is a problem supply in regard to the development of bioactive compound from marine biota. Therefore, it is very important for us to better understanding on the potential of marine microbe from our marine environment. And they will be benefit not only for human or us, but also for the ecosystem health. And at least we can work and at three different aspects. Number one is biodiversity. Number two, chemodiversity and biological function. Number three is biotechnology and application of the bioactive compound. This is a brief information about the Indonesian marine bioactive compound that has been carried out and developed so far. And you can see here, there's many potential for the application in the health field has been very significant. As you can see here, based on the work of Hanif et al. He reviewed the potential of Indonesian marine biodiversity. And you can see here, there is also a very significant contribution, especially from the sample that was collected from the eastern part of Indonesia. I believe that there are so many things, especially the bioactive compound, have not been detected yet. So we believe with the current situation, we will be able to develop and more marine drugs will be developed. Of course, our study and understanding on the microorganism, marine microorganism, despite the increased numbers of the marine microorganism, we believe that there is only approximately 1% of the total community of the microbe have been developed and explored. So more and more marine microorganism is waiting to be discovered. Therefore, we need a complementary technique, which is known as the metagenomic study, or the study of genetic material recovered directly from environmental sample in the marine domain is increasing rapidly. And today, marine metagenomic is used in a range of scientific and industry, from monitoring pollution, recycling water system, to disease surveillance, as well as the development of functional feed to aquaculture and bioprospecting. Marine metagenomic portal was made online in 2017. It is maintained by the Center of Bioinformatics at the UiT, the Arctic University of Norway. As you can see here in the slide, there is no single dot representing the marine metagenomic study in Indonesia. It will open the opportunity for us to explore the unexploited potential. And I very much believe that the lecture from Professor Russell will be our inspiration, especially to explore this potential that remain undeveloped so far. Now, please allow me to briefly mention our National Research and Innovation Agency, dubbed as BRIN, that BRIN has declared as a hub for multi-party collaboration. That's mean all infrastructure within BRIN is an open infrastructure, including human resources and facilities. We charter the need of the university researcher as well as the graduate student, as well as the need of the private, including domestic and international. And just recently, BRIN launched a program, what we call it a global platform. For example, global platform for biodiversity and global platform for climate change. We also have developed several strategic infrastructure between 2020 and 2024. This will be open to our partners in the national as well as the international level. This is an example of the infrastructure especially designed to cater the needs of research on biodiversity. It is open to public, industry, and other institutions. And we are conducting bioprospecting to support independence of product through biotechnology, providing genetic information and its derivative based on Indonesian bioresources, as well as meeting the needs of current technology, stimulating Sharia product, including halal food, pharmaceutical, and cosmetic. And this is another facility just recently inaugurated. We call it Marine Bio Industry Center in Lombok, which is located next to the very famous tourism area, Senggigi. And there, it is open also for international partner as well as for the national partner. It is well equipped with seawater circulation system as well as some state-of-the-art of the equipment. BRIN also established what we call it the National Fleet Unit. At the current time, we have organized 5 different research vessels that is open for anyone in the national, regional, and international level. For 2022, we have allocated 500 sailing days that can be used by either university lecturer or researcher, and also our regional and international partner. In the term of research talent and mobility, we have 4 different programs. Number 1 is Research Powerhouse. That means we have provide open access laboratory and infrastructure. Also, we have developed industry-driven based collaboration as well as competitive ecosystem and talent pool. For the collaborative aspect, we have provided several fellowships for the postdoctoral and then research fellowship, industry internship, diaspora engagement, as well as the startup program. For nurturing, especially the new generation of scientists, we have supported and provided fellowship for the program research assistant, visiting professor, as well as internship for lecturer and researcher, both in the national and international level. We try also to create critical mass through scholarship assignment, as well as a degree program by research and non-degree incentive, including also the capacity and competency building. So this is a good opportunity for our partners in the university and national level, as well as for the international level. As my concluding remark today, much of nature treasure trove of small molecule remain to be explored, particularly from the marine and microbial diversity to fulfill the human needs. And it is my great honor welcome Professor Russell to give his inspiring lecture today. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. So I underline what Professor Oki said, that metagenomic research in Indonesia is still scarce compared to other parts of the world. And hopefully Prof. Russell today could inspire us to boost the research in marine genomic biodiversity. And the second, that BRIN offers research facility actually to learn about marine genomic biodiversity in Indonesia. Invite national and international partnership. Thank you, Prof. Oki, and to audience. Now we are hoping that we can share photo session. So because now the participants are already reached 100, so it's time for photo session. Please turn on your video and our admin will, Miss Fitriani, could you please take a picture of us? Yeah. For the first slide, 1, 2, and 3, smile. Next slide, 1 and 3. Ya? Sudah semua ya, Mbak? Sudah 3? Oh, one more. Okay. 1, 2, 3. Thank you very much. And now we are reaching the core of this event. In a short while, we will start the general lecture by Professor Russell Hill. Let me read the CV of our guest professor. So I would like to share his CV. Yeah. Okay, so actually 60 pages of CV, so I could not read all of this, Prof. Russell, but briefly I can tell that Dr. Russell T. Hill received his PhD and honors in biological science at University of Natal, now University of KwaZulu-Natal, Durban, South Africa, and his PhD was from the University of Cape Town, South Africa. Dr. Hill began his work with the University of Maryland Biotechnology Institute in Baltimore, and promoted to professor in 2009, assumed his present position as director for the Institute of Marine and Environmental Technology, or IMET, a research center of the university system in Maryland. Dr. Hill is a marine microbiologist who has received numerous academic awards, including the University System of Maryland Board of Regents Faculty Award for the Excellence in Mentoring. He is one of 50 Maryland leaders to graduate from Leadership Maryland's 23rd class in 2015. He was elected the fellowship in the American Academy of Microbiology, the honorific leadership group within American Society for Microbiology. He was fellow of the Society of Industrial Microbiology and Biotechnology in 2003. Dr. Hill has served on the board of the International Marine Biotechnology Association since 2007 and was president of the association. The major focus of Dr. Hill research is the investigation of symbiosis between bacteria and marine invertebrates, in particular spawns. His laboratory was amongst the first to characterize the complex bacterial communities associated with marine spawns by 16 rRNA gene sequence analysis. His work on more than 20 spawn species around the world has contributed to the understanding of this diversity. The Hill Laboratory has elucidated the role of symbiotic bacteria in nitrogen and phosphorus cycling in spawns and the surrounding coral reef environment, as well as signaling process of these bacteria. The Hill Laboratory works on culturing many previously uncultured symbiotic bacteria and studying their role in production of bioactive compounds found in marine invertebrates. Another important research focus is the interaction between bacteria and microalgae that have potential in biofuel production. The Hill Laboratory is investigating the diversity of bacteria associated with microalgae, the effect of these bacteria on the quality of biofuel produced from the microalgae, and the positive and negative effects of the associated bacteria on the growth of microalgae. Professor has published hundreds of high-impact papers with 47 Scopus Index. So students, Professor has 47 among hundreds of his impact papers, which is each of these 47 papers is cited at least 47 times of other high-impact articles. So may I invite Prof. Russell Hill to deliver his lecture. Please, Prof.
Well, thank you very much, Dr. Etheka, for that wonderful introduction. And thank you, Professor Rajasa, for your really, really important opening remarks that highlight the huge potential of the whole field of marine biotechnology for Indonesia. And I think the value of the biodiversity of Indonesia to Indonesia and to all of humankind is huge in terms of the potential for discovering new drugs as well. So It's an absolute honor for me to be able to talk to you today. I thank all of you for attending this talk and for the wonderful welcome you've given me. I also really would like to thank Dr. Dewey Zilder for helping to bring us together and for her very productive 6 months that she just spent working in my laboratory as a Fulbright scholar. So with that, let me share my screen. Are you seeing my slides?
Yes, it's clear.
Very clear.
Okay.
Yes, it's clear.
Okay, good.
Yeah.
There we go. So, as Dr. Ethica highlighted, I will talk to you today about marine drugs from the sea, the genetic engineering of marine bacteria for health applications. And I will discuss a lot of the work done in my lab at the Institute of Marine and Environmental Technology, shown on this first slide. The IMET is part of the University System of Maryland, and we are very fortunate to have a wonderful facility right on the harbor in Baltimore, in a great location with a lot of beautiful labs, 42 labs in the building altogether. And I think it's really worth highlighting the fact that I will discuss a lot of work over quite a few years. And as we go through that work, the molecular approach has become more and more important. And Professor Rajasa really mentioned that, emphasized that with the potential of metagenomics to move forward the field of drug discovery from the marine environment. So to show you an overview of the talk that I will present to you, I will start off with a brief introduction of the process of drug discovery and highlight why the marine environment has so much potential. And I will then discuss some success stories. As Professor Rajasa said, we are no longer in the preliminary phase of this research. We now have some successes to report, and that is very important. I wrote an article, a commentary on this a decade ago with Professor Fennicle from Scripps Institute of Oceanography, and we called our article Marine Drug Discovery, The Tide is Turning, because we really are now seeing successes coming through. Now we'll discuss several of those successes, starting with a great story from a marine sponge that was investigated by some researchers in New Zealand. I will talk about our current work on actinomycetes isolated from marine sponges, discuss an important compound that has become a drug from a tunicate, and I will end with some of our most recent work, very exciting work that has taken more than a decade, but was finally successful in working out, using a metagenomic approach, working out the true source of some important anti-cancer compounds from a marine sea slug found in Indonesia. Sorry, it's found in Hawaii. So that is the overview of my talk. And I will start with a few remarks about the process of drug discovery. And it's worth reminding ourselves, this is a huge industry. In fact, the latest number available from 2018 estimated this industry at well over $1 trillion in worldwide pharmaceutical sales. That will have gone up a great deal during the pandemic. So we are talking about a huge industry with very big companies involved. And several of the companies that I've listed here have sales of over $50 billion for each of these companies per year. One of the companies, Pfizer, invests around $8 or $9 billion each year in research. And to put that into perspective, that is more than the research budget of the National Science Foundation, which funds most of the fundamental research in the United States. So one company is spending more on drug discovery than the National Science Foundation is spending in other research areas. Why are these huge expenditures so important? Well, this is a difficult process and it can take a long time. It's been estimated that it often takes over $1 billion to discover and develop and take to market a single new drug. This is why we have to be realistic when we are doing the early stages of drug discovery and investigating biodiversity in Indonesia or in an African country. I've done a lot of work in Madagascar, for example, or here in the United States. We are doing the first step, and we will have to have a lot of collaborators and a lot of partners and a huge investment to have a good chance of actually ever getting a new drug onto the market. The payoff can be huge as well for these companies, and there are currently around 100 blockbuster drugs, billion-dollar drugs. And those drugs bring in around $1 billion every year for the life of the patent on the drug. So that's to just remind us what a huge industry this is and what a challenging process. So what is the process that is followed? Well, there are a number of different approaches. And traditionally, when we are discovering new compounds from natural sources, we are taking an approach that has been called grind and find. We are taking many different samples. We can be accessing marine biodiversity, sampling hundreds of different sponge species, ascidians, other organisms that have a good track record at producing interesting compounds. We can also be isolating many different bacteria, originally mainly from soil samples from all around the world, more and more now from marine bacteria, and growing up those bacteria, fermenting them and making extracts. And once you have those extracts, you go through many screens. The more screens your extracts go through, the more chance of success, until you find some important extracts which you then have to investigate to isolate individual compounds and look at the activity of those compounds. So this is a laborious process, a long process, but it has been a hugely successful process, and it's worth remembering that. Pharmaceutical companies more and more now take other approaches. They try to design drugs from first principles that will target a particular receptor. Or they take a known compound and by combinatorial chemistry, they produce many hundreds or thousands of related compounds similar to the known compound and look for an improvement in the performance as a drug. And these approaches were regarded as very, very important 20 or 30 years ago. Many companies are still taking these approaches. The payoff has not been as big as we expected. And there's a resurgence, a return to studying natural products as sources of new drugs. And in particular, of course, we are interested today in marine natural products. So there are a lot of advantages. There's the potential to discover entirely new classes of bioactive compounds, compounds that no chemist would ever dream up. And there's a successful track record. It's worth remembering that more than 30% of the current therapeutics that are used in medicine derive from natural compounds. And when you look at anti-cancer compounds, that percent is even higher. So why the marine environment? Well, I'm going to keep this very short, and we had a great overview of this by Professor Jaza already. I just want to make a few quick points. When we think of the marine environment, and those of us who have been fortunate to snorkel and dive around the world and in the most spectacular environments in Indonesia, you're used to seeing wonderful scenes under the oceans with a huge amount of biodiversity, with many sessile marine invertebrates, such as the sponge shown in the middle of this picture, and there are other sponges around here and many other organisms as well. And one striking point to make is that there is huge biodiversity, but also that these organisms are all in competition. And the sponge you see in the middle of the picture is not covered by fouling organisms. The alga around here and other organisms have not grown over the sponge. And the reason often is that many of these marine invertebrates are chemically defined. They are producing a huge array of highly potent compounds that are toxic to other organisms. It's not unusual for a sponge to have tens or hundreds of potent compounds associated with the sponge, inside the sponge. So this is a very good place to be looking for new drugs, and in particular anti-cancer drugs, because if you have highly toxic, highly bioactive compounds, those compounds have a higher possibility of being toxic against rapidly growing cancer cells. So that has been a particularly promising area from the marine environment. And I said earlier already that there is a record, a successful track record. And I want to just highlight some of that. Right now, we have 10 approved drugs from marine sources that are used as pharmaceuticals. This is building on a long history. From the 1950s, some compounds were identified from sponges in the Caribbean, nucleoside compounds that inspired chemists to make similar compounds that turned out to be some of our very first antiviral and anti-cancer drugs. So with that long history, we've built on that, and there are now 10 compounds, drugs on the market. And you will see of the 10 that I've listed here, 8 are anti-cancer compounds. That gets back to the point that I made about the marine environment being especially promising as a source of anti-cancer compounds. I want to point out this web link at the bottom. This is a really good resource for tracking the progress of many different drugs from marine sources. And it's maintained by Alex Mayer at Midwestern University. So that's an important resource for anyone interested in bioactive compounds from the marine environment. Okay, so now let me move to my favorite organism, sponges, and tell you a little bit about sponges. The story that I want to tell you here is work done by 2 great scientists working together in New Zealand. And this led to a very important drug from the marine environment. So this compound, you can see the complex structure, this helicondrin B compound, was discovered many years ago, actually found by several different groups in different sponges at different sites around the world. And that's an interesting point. Whenever you see that happening, it creates the interesting possibility that the compound is not being made by the sponges, but possibly by the bacteria that live inside the sponges. Because you have different sponge species containing the same molecule. One explanation sometimes is that they actually have the same bacteria living in different sponges that are making the compound that you're interested in. So this compound was discovered, found to be extremely potent as an anti-cancer compound, especially against breast cancer. The huge problem, and an important point I want to make here, is that for the first 10 years of knowing about this compound, the worldwide supply of this compound was 300 milligrams. And to get that 300 milligrams of helicondrin B took a collection of 1 ton of sponge. This is a rare deep water sponge from New Zealand. So divers had to collect 1 ton of the sponge to get 300 milligrams. Because this was such an exciting compound, the National Institutes of Health funded a sponge survey. That survey found that there were only 289 tons of this lysodendric sponge on the planet. So right away, you see the problem. 1 ton was collected to produce 300 milligrams. Collecting the sponge cannot be the answer to moving this drug forward because the supply is so limited. So this is a problem that comes up time and time again when we're studying marine organisms and bioactive compounds and trying to move them forward as drugs. We hit the supply problem. I will mention this again in several other cases. This compound could easily have died because it was impossible to get enough for it to be marketed as a drug. But fortunately, a very good Japanese pharmaceutical company got involved, a company called Eisai. And this company worked with the 2 scientists who discovered this compound, Murray Monroe and John Blunt. And the company, Eisai, discovered that in this case, they could synthesize part of the molecule. They did not have to have the entire molecule. By synthesizing part of the molecule, they retained very good activity, and the compound could move forward as a drug. And this compound, as the drug started off with one name, but now while it's marketed, it's called Halaven. So a very successful story, and it makes several points. Sponges can be a good source of compounds. This process can be difficult and challenging. You can hit a supply problem, and there are different ways to overcome the supply problem. Sometimes you are fortunate and chemists can overcome the supply problem. In many cases, actually, you need the whole molecule, and synthesis can be impossible or impractical. So you have to find other ways around the supply problem. And that is where our metagenomic approaches can be so helpful. Okay, so that's an introduction to something about sponges. I will keep talking a little bit about sponges, but now I want to talk about sponges in the context of the bacteria that live inside the sponges, and especially my favorite bacteria, Actinomycetes. These are bacteria that I worked on for my PhD, and I developed a genetic cloning system for a genus of Actinomycetes long before I did any work in the marine environment. But now, by working on Actinomycetes and sponges, I'm working on my favorite microbes and my favorite marine invertebrates at the same time. So I started this work in Australia on the Great Barrier Reef, working with a very talented PhD student, Nicole Webster, who's gone on to be one of the best sponge microbiologists in the world. And we worked on many sponges. Something we learned early on is that sponges are often crammed full of bacteria. And many sponges, 30% of the weight of the sponge or more is made up of the bacteria. So when you find an important compound from a marine sponge, you really have to be thinking about the bacteria as well. Often it is the bacteria that are the true source of the compound that you are interested in. I already mentioned that that is likely in the case of Helicondron B and the sponge Lesodendrix. And we found that to be the case in many other sponges. That's led us to study bacteria associated with many different marine sponges around the world. And these two sponges we've studied in great detail. These are from the Florida Keys. One other quick point to make here, one of the nice things about working on sponges is you can take a sample, you can cut part of the sponge and leave the organism where it is. This was sampled here, and those sponges recover very well. So you're having no impact on the biodiversity, and you're still getting the samples that you need. And of course, if you're able to cultivate bacteria that are making important compounds, that's a good way to overcome the supply problem. So now I want to home in on one sponge that I'm going to talk about in a little bit more detail. And that is the beautiful sponge shown in the middle of this picture. This sponge is called Zestospongia muta, the giant barrel sponge. And it's the biggest sponge in the world. It can grow over 2 meters high. And you have a very similar sponge in Indonesia called Zestospongia testudinaria. They are different species, but we actually compared the bacteria in both sponges and found that the bacterial communities are very similar. So there's a lot of interesting work to be done comparing the microbial ecology of these two sponges. And that work I'm not going to get into today. That is all published for anyone who's interested in looking at that further. What I want to focus on today is that we have used Zestospongia muta as a source of many different important bacterial cultures. So I believe this is the sponge that is best studied in terms of its microbiology. We've done extremely detailed 16S ribosomal RNA community analysis of the sponge, but more importantly, in many ways, we have succeeded in culturing over 600 different species of bacteria from this one sponge. So a single sponge species can be a huge resource for microbiology. And it's worth thinking about that. Every time we lose a sponge species, every time a sponge becomes extinct, we are also potentially losing many different bacteria that are tightly associated with that sponge species. So we've done a lot of work on Zestospongia muta. We've isolated many, many different groups of bacteria. But for today, I want to focus on the actinobacteria. And this image shows some of the beautiful actinobacteria that we've isolated from sponges. At this point, much more than 20 different genera. Actinobacteria are particularly important for drug discovery because this single group of bacteria is the most important group in terms of making important bioactive compounds. And as one example, around 70% of all the antibiotics in clinical use come from actinomycetes. Many of those are from soil actinomycetes, from Streptomyces in particular, which historically was a very important genus for discovery of antibiotics. By going to the marine environment, we are finding new actinomycetes that nobody has screened before. And in that way, we can find novel compounds. And I want to tell you just one story, ongoing work in my lab. And the graduate student working on this project, Daniela Tazabi, has done a great job in looking for actinomycetes from sponges that make anti-tuberculosis compounds, anti-TB compounds. So I'm going to take a minute to talk about TB. So this disease is caused by the bacterium, Mycobacterium tuberculosis, which happens to be an actinomycete itself. It's a huge problem worldwide. Around a quarter of the world's population is infected with this bacterium, even though in the vast majority of cases, those are latent infections. But there's a growing concern, especially in immunocompromised people, where TB starts to spread and there's more and more development of antibiotic resistance. So there's a huge problem. And this is a current issue. Because of COVID-19, TB is on the rise. Great progress was being made in treating TB, but a lot of those resources have been diverted because of the pandemic, and TB has become more prevalent over the last 2 years. So there's a lot of concern about this disease and a huge need for new drugs to treat TB. And we are working on a number of different possibilities here. I'm going to give you an overview of our approach. We started off with a fairly small collection of highly novel actinomycetes, around 101, 58 of those we've grown and screened very carefully at this point. And of those 58, we found 11 that showed really good activity against Mycobacterium tuberculosis. So that's encouraging. Now, normally you would do a whole lot of extractions and get chemists involved to try to figure out those compounds. We are doing that, but in parallel, we are taking an intensive genomic approach. So these 19 actinomyces strains, we've now sequenced very carefully, and Daniela has done an excellent job of comparing 3 different assembly bioinformatic pipelines to get very good assemblies of these 11 actinomycete genomes. And she's mined those genomes very well, looking for biosynthetic gene clusters, BCGs, that may be making some of the compounds that we are seeing give the anti-TB activity. While all that's happening, we are also working with a chemist, an old friend of mine, Leva Rakondrabi, who's originally from Madagascar. I met Leva when I was doing some work in Madagascar many years ago. His PhD is from Japan. He has a postdoc from a top lab in the United States doing natural products research. And he is now an associate professor at Ohio State University. So he's a wonderful collaborator, and this work is moving forward in a very interesting way on many fronts. And I want to tell you just a little bit more about part of that. So one of the organisms, one of the 11 that we found to be very interesting is a Micrococcus species. This strain shows potent activity against Mycobacterium tuberculosis with this large zone of inhibition here. It also shows activity against some other strains of Mycobacterium, Smegmatis and Marinum. So we are focused on this, and as I outlined, we are taking a dual genomics and chemistry-enabled approach to identify the compound that's responsible for this promising anti-TB activity. This is ongoing work. It's at an exciting stage. We have very good closed genomes. Most of our genome sequencing for the molecular biologists in the audience. We do Illumina sequencing in-house at our institute, but we often also send out for some PacBio sequencing to help get better closure of the genomes. In the case of many of these actinomycetes, this can be quite challenging because the genomes are very large and there's a lot of repetitive DNA. That was not the case for this organism. The Micrococcus has quite a small genome, only around 2.4 megabases. And we know from the genomic analysis of these genomes that this strain is likely to be producing some carotenoids. There is also a hint that it's producing compounds related to some compounds that have previously been known to have anti-TB activity. But this relationship is not strong. It's given us a clue and it's helped us guide some of the chemistry. This is an ongoing story, and we hope soon to have some exciting publications of a novel compound from this organism. And I'll tell you, Daniela is getting near the end of her PhD. This is just one of many projects she's working on, and she is going to continue in the field of marine natural products discovery. So she will be, I think, doing interesting work in the future in this area. So here's Daniela again. I've told you about the Micrococcus. Of course, it's always interesting to do a phylogenetic analysis, because that can give you some clues about what is going on. And when we did our phylogenetic analysis, we found that we had 3 different Micrococcus species that are close together, that are making something potent, and we'll be able to tell you more about that soon. But in addition to this, these marine Micrococcus species are closely related to some other marine Micrococcus, including a strain that I isolated more than 20 years ago that we worked on together with Merck and with a foundation called Medina. And this is old work. I'm not going to get into details on this, but that led to the discovery of a new antibiotic called Cocurin. So it's encouraging to me and very interesting that the Micrococcus that we're looking at now are quite closely related to other marine Micrococcus, including one that we know has already given a new antibiotic. So that's part of the reason for doing the detailed phylogenetic analysis. So that covers what I want to tell you about sponges and actinomycetes. But I've told you that there are many interesting compounds moving forward in the marine environment. And I want to give you an example from work from other labs and also from industry on a really important compound that came from a tunicate. And this is, of course, this is an area that's really worth looking at in Indonesia, where there are a lot of novel tunicates that are not found anywhere else. I think this is an important group of organisms where a lot more research needs to be done. So to make that point, I'm going to tell you the story of a compound that started off being called actinosidin ET743. So first of all, just a code name, ET743. Then the chemists gave it a name, actinosidin. And then it moved forward and it became a successful drug. And then the marketing people give it another name, Yondelis. And we often see that happening as compounds move through the discovery process in the marine environment. So this compound is an alkaloid. It comes from a Caribbean tunicate, this beautiful little tunicate shown here, that grows on the roots of mangrove trees. And it is fairly sparsely distributed. So it was a real problem in the early stages of discovery of this compound, because the compound had to be collected from the wild, and it was having a major impact on the compound. It was being collected at many different islands in the Caribbean and affecting the population. Fortunately, that problem was solved, and I'll tell you how. But this compound, the actinosidin, this is the structure here, has very promising anti-cancer activity against several different types of cancer. And it binds to the minor groove of double-stranded DNA. So this has moved forward and is on the market as a drug in Europe. It's moved forward by a series of steps with the involvement of a great company called PharmaMar, a Spanish company that does very important work in the field of marine drug discovery. In this case, the first step was actually to grow the organism in aquaculture. So that's another good option, another thing that's, I think, very worth considering for promising organisms in Indonesia. because you can get things more under control. You don't have to collect organisms from the wild, and you reduce the ecological impact that we always have to be thinking about. So a small company started up called CalBio Marine. They grew Echinocida in aquaculture and managed to get large biomass. They would start off with 1 kilogram of the organism collected from the wild, and they would get that up to 50 or 60 kilograms in 60 to 90 days. So this was very important to get enough of the compound for companies to become interested in it. And PharmaMar then became interested, and this whole story took a very strange twist, because PharmaMar realized that the Echinocida 7043 compound had some similarities to a compound from a completely different source. This is a compound made by a terrestrial bacterium that grows on plant leaves, a compound called Pseudomonas fluorescens, a bacterium called Pseudomonas fluorescens. And PharmaMar had very good chemists. They realized that this bacterium, which is very easy to grow in fermentation, they could use that system to make this compound as a precursor and then do a 21-step organic synthesis, still challenging, but economic, and make the compound identical to the one that was discovered in the marine organism. So there are a few points here. Once again, chemists are overcoming the supply problem, but they're using a bacterium. And this gives a very good clue that this compound, which is made by a bacterium and is related to this compound, it implies that this compound is probably also being made by a bacterium, not by the tunicate, but by a bacterium that's symbiotic with the tunicate. And after a lot of very good research, and you can read more about it in the paper that I listed down here, that was found to be true. The problem was the symbiotic bacterium was identified by metagenomic approaches, but it was never successfully cultured. Nobody has yet worked out how to grow this bacterium. So in fact, the pharmaceutical is still made, Yondelis is still made by the original process using the Pseudomonas bacterium. However, this is another reminder that we should always be thinking about the symbionts, because it's often the symbionts that are making the compounds that we're interested in. And that was the case here. This organism does not have a lot of symbionts, but it still took quite a while to figure out that there was one key group of symbionts, and the entire biosynthetic pathway from the symbiotic bacteria has now been cloned and characterized. However, it's still easiest to produce the compound of interest, the Yondelis, using the terrestrial bacterium that I told you about. So the Yondelis is still produced by semisynthesis. Cyclosaffricin is produced by fermentation of the bacterium from plants. The 21-step organic synthesis gives an identical structure to the structure that was first identified from the tunicate from the marine environment. So you get the sense of how difficult this can be to move a compound forward from the first point of discovery and to overcome the supply problem and to actually get the compound to move forward as a new drug. And I want to end with the last story where we have been successful in doing something like this by using a metagenomic approach. And once again, this has not been easy. This project has lasted for around 10 years, but we finally have a very good understanding of the compound that we are interested in. And the organism that I want to tell you about is a saccharoglossum, a sea slug, another organism where Indonesia has greater biodiversity than anywhere else on the planet. But in this case, this saccharoglossum is actually found in Hawaii. It's widely distributed in the Pacific. It's solitary. It lives by itself usually, and it's almost impossible to collect because you can't find enough of it. But for 2 or 3 months of the year, this saccharoglossum moves to Hawaii. And I'll tell you more about that in a minute. But the other point I want to make, the saccharoglossum is a very interesting invertebrate. It lives on a single species of alga called Bryopsis, shown in the background here. And the saccharoglossum is sap-sucking. It sucks the sap of the saccharoglossum, and it keeps the chloroplasts. And the chloroplasts are kept under the skin of the marine invertebrates where they photosynthesize. Photosynthesis continues. So once the saccharoglossum has had a good meal and it's captured these chloroplasts, it can live for several months by photosynthesis, relying on kleptoplasty. It has, klepto is the Greek word for steal. It has stolen the plastids from the alga. And one interesting possibility that we were considering was whether we also have a case of kleptochemistry. In addition to stealing the chloroplasts, we believe that the saccharoglossum may be stealing some chemistry from the alga. So let me tell you more about that. This is the beautiful organism that we're talking about. This is in captivity at our institute. And the compound that we are interested in is this compound called Kahalide F. Kahalide F has some very promising anti-cancer properties. It has not moved forward as an anti-cancer drug because of the supply problem. Pharmaceutical companies do not want to invest in this compound yet because there's no reliable supply for the compound. So the compound was discovered by the grind and find approach that I told you about right at the beginning, actually by a friend of mine, a very good natural products chemist called Mark Hammond, during his PhD 30 years ago in Hawaii. He collected this organism, he ground it up, made organic extracts, and discovered very promising anti-cancer activity. We wanted to move forward and see if we could understand where the compound is coming from. And there are lots of possibilities. The compound, interestingly, is found at high concentrations in the saccharoglossum, up to about 1% dry weight, and very low concentrations in the alga. So right away, that raises the possibility that the alga could be making the compound and the saccharoglossum could be concentrating it, bioaccumulating it. There's another possibility. The saccharoglossum may be making the compound and it makes a lot of slime and crawls over the alga. And it could be leaving a trace of the compound that chemists then find on the alga. And the other possibility that really interested us is that it's not the saccharoglossum or the Bryopsis that make the important compound. It could be a symbiotic bacterium. And that bacterium could be growing in the saccharoglossum or in the alga. So it's a complex system. It took us a long time to work this out, but it was a great project. We got to work at some beautiful places. This is the small enclosed bay in Hawaii where this saccharoglossum comes to mate. So for 2 months of the year in the spring in Hawaii, you can find lots of the saccharoglossum in this one place in the entire world. you can wade out here and you can collect the Saccaglossum easily. So that was where we did our collections over many years. This project has taken us well over a decade. We would collect here, and then we would go to the Hawaii Institute of Marine Biology. A good friend of mine, Joanne Leong, was the director there. And I have other colleagues there who very kindly gave us lab space to work on this interesting project. And you can see we are working in some beautiful places. So the student who worked with me on this project is shown here, Jeanette Davis, and she has been wading out. And we don't even need to scuba dive here. This is shallow water and the Saccaglossum is very abundant. So we can collect the Saccaglossum and the green alga simply by snorkeling. And we did a lot of different work. We did culturing of all the bacteria that we could from the Saccaglossum, the Alecia rufescens, also from the alga. We did fluorescent in situ hybridization to try to study the bacteria. We did chemistry in these different organisms. But what really cracked this open and solved this problem after many years was some very good metatranscriptomic and metagenomic work. And this was done by a friend of mine at Princeton University, working with a postdoc who did his PhD in my lab and then moved to do his postdoc at Princeton. So it took a huge amount of money and a lot of metagenomics to work out this really complex system. And we came out with a great solution here. We really have a deep understanding of this. And Jindong Zhan is the postdoc who did his PhD with me, who is first author on the paper that describes the outcome of this project. And that is all explained here. If you look at this figure, you have the Bryopsis, the green alga, shown here, and you have the sap-sucking sea slug, the Alecia rufescens, shown here. And the metagenomics taught us that there was a bacterium present in the green alga, a very novel bacterium that has been described as a new genus and species. And this bacterium will be absolutely impossible ever to culture. It has a highly reduced genome. For example, it cannot make even a single amino acid. So this is a bacterium with a very reduced genome. However, 20% of the genome of this bacteria is dedicated towards making the compounds that we're interested in, the K-halide compounds. So the bacterium is present as a symbiont, totally dependent on the green alga. It gets all its nutrients from the green alga, but in return, it makes these potent compounds, the K-halides. And there is a whole suite of K-halides, different types of K-halides. And it's the K-halide F that has the most promising anti-cancer properties right now, but we believe that this is an important resource for drug discovery because there are many other K-halides present. The bacteria makes the K-halides. They protect the alga. The K-halides also have antifungal activity, so we think that they protect the alga from fungal attack. And then the Saccaglossum sucks the sap, it steals the chemistry, and it steals the chloroplast, and it greatly concentrates the chemistry. So the K-halides become highly concentrated within the Saccaglossum. So we have an obligate symbiont, a novel obligate symbiont that could only be discovered by this metagenomic approach, that cannot make any of its own nutrients, but dedicates 20% of its genome to making these very important bioactive compounds that were originally discovered in the saccharoglycan that feeds on the green alga. So I think this is a great place to end. It shows the complexity of marine symbioses. It also shows the fact that you should, when you discover a promising compound from a marine invertebrate, you always have to be thinking about the microbiology as well. You have to be thinking about the possibility that the compound is actually being made by a microbe. And to solve some of these problems, you have to understand a lot about the marine invertebrate, its lifestyle, what it's feeding on, where it goes. All of these things can give you clues to help work out these interesting and complex puzzles, and ultimately to help drugs move forward in the drug discovery process. So we are very happy that we succeeded in doing that with the story with the Elysia rufescens. It makes it more likely that kahalides will one day be useful drugs that are actually used to treat cancer. And in particular, kahalide F shows promising activity against pancreatic cancer, which is one of the most devastating types of cancer for which there are already no effective therapies. So this is an exciting and ongoing project. And to summarize some of what I've been talking about, I've emphasized the challenge of the supply problem. So many important compounds are discovered from marine organisms, and they never move forward because there is not enough of the compound to give pharmaceutical companies the reassurance that they need, that they will really be able to bring the compound to market. So unless you can overcome the supply problem, the compound is going to die. It's not going to move forward as a drug. Microbiology can provide solutions, and we need to take combined chemistry, microbiology, genomics, and metagenomics approaches. I think this is an area with huge potential in Indonesia, considering your fantastic marine biodiversity and the very strong marine natural products chemists in Indonesia, and the growing strengths in microbiology and metagenomic approaches. So Indonesia could really become a leader in this area. And marine natural products has the advantages of revealing entirely new classes of bioactive compounds that can become new drugs. And I want to end by just thanking and acknowledging the many different graduate students and postdocs who've worked with me on various aspects of marine microbiology over the years. My lab does a lot of work in marine microbiology in addition to the drug discovery, in particular looking at the role of marine sponges in nutrient cycling in the coral reef environment. And I will end by thanking all of you for your attention. I thank you for this opportunity to talk to you, and I'm very happy to take any questions that you may have.
Thank you very much, Prof. Russell Hill. Very exciting lecture. I would like to summarize some points from the lecture that Prof. Russell has shown us successful drug development from marine environment. For example, halaven as breast cancer drug, Yandala, kahalide, yes. And among his favorites microbe is actinomycetes, Micrococcus, and Pseudomonas. And also that today the supply problem of drugs allows us to actually try to find the bioactive compounds, and metagenomic approach will help us to find out where the concentrated bioactive compounds is. And also actually preliminary study about bioactive compounds in the marine environment requires collaborations, large collaboration among countries. And also, finally, more than 30% of drugs developed today actually is from natural compounds. Okay, so Prof, there are several questions that I can record from the chat room. First is from Nur Hayati, so acting one by one. What is the media used to inoculate the actinomycetes? How long these actinomycetes can grow? Probably from the sea to the land. What is the method for analysis the active compound, and how about the standard or library regarding active compounds from actinomycetes? Okay, Prof, please just respond, and then we will move to next question after this.
Sure. Well, there are lots of good questions here, and I will tell you many of our secrets quickly. And some of them are not so obvious. So in terms of the media, a classic medium that we use very often is a standard actinomycete medium called ISP2, International Streptomyces Project 2. So if you just Google ISP2, you'll find that medium. We simply make it up and add 2% NaCl to that medium. So that is a medium we use often, a whole lot. But if you want to really explore the actinomycete diversity from marine invertebrates, it's worth using quite a range of different media. And in some of our studies, we use up to 10 different media. Much of that is in papers we've published, and you'll get all the details on those other media that we use. But actually, your next question is maybe even more important, and it's not obvious. How long do you take to isolate these? So this is something that still puzzles me, but we've discovered a trick that works very, very well. What we are doing when we isolate marine actinomycetes is we streak out the plates. We include a couple of antibiotics to inhibit fungi and to inhibit especially fast gram-negative bacteria. So we include nalidixic acid, nystatin, and cyclohexamide. And you'll find all these details in our papers. But what we then do is we seal the plate with parafilm so it does not dry out. And we incubate them for at least a week. And then we check them carefully and we mark any colonies that have come up. And you soon start to recognize actinomycete colonies from their morphology. And then we put the plate back and we incubate it for another week and check again. And some of those colonies will have got bigger and we'll subculture them. But the trick is to seal the plate again and put it away for a month or 2 months or 3 months and keep checking. And something remarkable happens. We've marked the colonies that came up early, and other plates on the plate, nothing grows. And then suddenly after 1 month or 2 months, a colony will appear. And you may think, well, that's really difficult. It's going to be difficult to ever work with this if it grows so slowly. But once that colony appears and we subculture it onto a new plate, it grows fast. It grows in a week or two. So it becomes manageable. So we discovered some of this by accident a long time ago, but long incubations can be hugely helpful. I know I mustn't take too much time in answering an individual question, but I'm reading the other things that you asked. What is the method for analysis for the active compound? Well, you know, we are microbiologists. In my lab, we're very interested mainly in looking for antibiotics because that's something we can do very easily. We make organic extracts with ethyl acetate. We evaporate them down. We resuspend in DMSO and we spot them onto little paper discs and we put them on lawns of the bacteria that we want to check for antibiotic activity against. So a very simple process and something we can do easily in a microbiology lab. But there are many, many other things you can do. And the best thing is to have collaborations with chemists or with pharmaceutical companies that are screening for lots of other activities, because then you can take your same extract and get it screened for many other important activities. And it's the chemists and the pharmaceutical companies that get into the next final part of your question on the standards and the libraries. And some of them, they have less interest these days in developing extract libraries, but they're very interested in isolating purified compounds and putting those in their libraries. So that's the quick answer to your very good questions. Thank you.
Okay, fine, Prof. So I want to inform the audience, if you want to raise hand and present the question in person, you can do that too. For the second question from Hayatun Fuad, this is our student. I want to ask Prof. Russell regarding what method is used to test the pathogenicity of the bacteria before being applied to health. Okay, please, Prof.
Sure, you raised a couple of interesting points here. First of all, we are isolating novel bacteria from the environment. The vast majority of these bacteria are non-pathogenic, but we never know that for sure. There's the possibility of isolating something from our sponges or from other marine sources that is pathogenic. So we always have to follow careful technique with all the work that we do. But in terms of the pathogenicity before being applied to health, in fact, the bacteria are never really applied, or people should not become exposed to the bacteria outside the lab, because you go through the many steps of making an extract and purifying the compound. And it is the compound that is developed as the drug. And that has to go through very, very strict tests through phase 1 and phase 2 and phase 3 clinical trials to make sure that there are no bad effects from the compound when it's used as a drug.
Okay, is that all?
Okay, Ezeka, I must ask you to please translate the next question.
Okay. Very clear, Prof. Thank you very much. And the next question, the third question is from Rifan Ahmad. Prof. Oki, so this question is for Prof. Oki. In Indonesia, mohon dijelaskan kembali skema kerjasama untuk komersialisasi antara BRIN Research Center dan pihak swasta. Prof. Oki, please explain the cooperation scheme, especially for commercialization between BRIN, the research center, and the private sector. Thank you. Please, Prof. Oki, would you please respond?
Okay, thank you. Number one, in 2022, BRIN has call for proposal for the all year round. That means you can apply anytime during 2020, and the review of the proposal will be conducted every 2 months. So that's one, the possibility. In this, you may apply to have a consortium with the university and BRIN, as well as with the private and also international partner. That's one possibility. The second one is that the university, Indonesian university, might apply for the establishment of research collaborative center. So the budget will be provided by BRIN, but the center will be established in the university. And this consortium should include also the staff from BRIN, as well as there's a possibility to include the private sector as well. So this is quite a big funding, and this will last between 3 to 5 years period. So I think it is a good opportunity also for the Indonesian university to, for example, you want to research collaborative center that include, for example, Professor Russell Hill, then basically you can apply. And the other one is that under the deputy for the utilization of research and innovation, there are several schemes, such as the establishment of a startup company, and also for the incentive for the utilization of research result. So it's depend actually in your needs. So as for the research grant, it's a free scheme in terms of you may for basic research apply research grant as well as development. So it's quite new system that I mean the possibility to get support, financial support can be in various form. So you may try different type of the scheme so that you will get support from BRIN. I think that's all from you.
Terima kasih, Pak.
Yeah.
Okay, so we are moving to the fourth question from Fazri. The extent of research on marine sponge. What bacteria are found in marine sponge? If sea sponges have great potential for antimicrobial drugs, will they not have impact on sponge population and marine ecosystem? I hope you understand the question, Prof.
Yes.
Okay, good.
Just give me a second. I'm just looking through the chat. Okay, I've got the question now. But just before I answer that question, I just want to point out that it sounds as though you have some fantastic opportunities through BRIN, and I really congratulate Professor Rajasa for setting up this very innovative way of funding, and especially having a rolling deadline every 2 months. I think that makes a whole lot of sense. And I don't know why we don't do that with all types of funding agencies. Anyway, let me get back to this question. What bacteria are found in marine sponges? Well, the diversity of bacteria in marine sponges is remarkable. It's very, very large, and it's now very well understood. In fact, the microbiology of marine sponges is understood as well or better as the microbiology of the human metagenome, the microbiome, and marine sediments or the water column. And this is the result of a huge project with about 50 different collaborators around the world and unlimited support for sequencing over the last 5 years or so. So we were fortunate to be able to sequence, do metagenomic analysis of the bacterial communities associated with more than 50 species of sponges around the world in huge detail. And look at the literature on that. There are some recent papers. It's a remarkable story. A couple of names to Google just to find the right papers. My ex-student, Nicole Webster, Webster, and if you Google Webster and also Thomas, Thomas, they are lead authors on some of these papers. So if you Google those two names and marine sponges, you'll get a huge amount of good literature. So, and I'll say a little bit more. It's very interesting. Different sponge species growing right next to each other have completely different microbial communities. But then the same sponge species growing thousands of miles apart end up having very similar bacterial communities to each other. So the bacterial community associated with a sponge is almost like a fingerprint for that sponge. There's a very close relationship. Even though these are really diverse communities, they are quite stable in a particular sponge over several years. and they are stable in a sponge species over long geographic distribution. So it's an interesting story. So, and your point about the potential for having an impact on sponge populations and marine ecosystem, that's a very important point whenever we are accessing any marine invertebrates. Fortunately, for sponges, if we are careful, we are usually able to just cut off part of the sponge. Even if you cut the sponge in half and take half the sponge as your sample and leave half behind, that sponge will regenerate and it will be okay, it will regrow. So sponges are easier than many other marine organisms. But yeah, the collection has to be controlled. It has to be done carefully. Otherwise, there are classic examples of this. There are very important organisms, cone shells, that make toxins that have biomedical potential. And there is a science paper pointing out that the populations of cone shells has actually been impacted by scientific collection. So it's something we have to be aware of, and we have to be careful with that.
Prof, there are two more questions. So from Nur Hidayati, Prof. Russell, in my thesis research, marine bacteria from stomach content of sea cucumber were reactive in the antithrombosis test from blood clots. The research will be very developed in the future. In this opportunity, hopefully, there will be a way to develop this research. Maybe me, Prof. Oki, and Prof. Russell can work together. Oh, sorry, Prof. This is the receiver of a master grant thesis in the period of Prof. Oki. So I think very excited, enthusiastic student. Please, Prof. Russell.
So I've followed some of the work on the bacteria from Holothurians, and I think it's a really interesting source of novel bacteria. It's an interesting area to be working in. So congratulations on your thesis findings. And I think there's a lot of potential. You never know. I think it's important to say, I'm not sure if you play the lottery in Indonesia. Some countries have lotteries, some countries it's not even legal. Trying to actually get a new pharmaceutical all the way onto the pharmacy shelves, for us as scientists, it's like buying lottery tickets. You have to buy many thousands of tickets and your chances of ever winning are small in terms of getting a new drug actually on the market. And I talked about this at the beginning of my talk. This is a long process, and the average cost now to get a drug on the market in the US is over $1 billion, and it takes more than 10 years. But there's so much interesting science to be done along the way. And it's important because if enough of us are doing this research, then we are going to find the new compounds, and especially new antibiotics. I'd encourage you to really think about antibiotic discovery. The problem of antibiotic resistance is getting worse and worse. Pharmaceutical companies do not put a lot of research into new antibiotics because they typically don't make as much money as other drugs that people have to take every day for the rest of their lives. For an antibiotic, you take the drug for 5 days or 10 days, and hopefully you get better if it's a good antibiotic. So I think academic researchers and small companies have a very important role to play in antibiotic discovery. But it's very interesting that you have some anti-thrombosis activity. That's the sort of activity that pharmaceutical companies are very interested in, because often those drugs need to be taken for long periods of time by patients and can make a lot of money.
Thank you for the encouragement, Prof. And the sixth question is from, who is this? My second question, Prof. Hill. It was very interesting point in the case of sea-slug symbiosis in finding out which is responsible, for bioactive production, whether symbiont bacteria or cyst-like host, or they do both together. Based on a metagenomic approach, how to make sure that it will be always the same pattern on different geographic or environmental conditions? Thank you.
So that's something that has not really been looked at in a whole lot of detail yet. We were very happy to figure out that there was this obligate symbiont present in the Bryopsis. It raises the interesting possibility that other species of Bryopsis around the world may have similar symbionts that make other novel compounds. And I know my collaborator, Professor Mohammed Donia at Princeton, is interested in looking at that further. So that's not work that my lab is continuing with, but Professor Donia, who he is continuing with that work. So that's a good question. There's one other question that I saw a little bit earlier in the chat that is very interesting. In the case of the tunicate aquaculture, this is a good question and very true. Often when you move organisms into aquaculture, you run into problems that they stop producing the compounds that you're interested in. In the case of the tunicate, that did not happen. However, the concentration of the compound decreased. So the tunicates did not produce as much of the compound once they were in intensive aquaculture, but they still produced enough of the compound to really help move it forward. So it's different in every case. I've done some other interesting studies moving sponges into sort of natural aquaculture systems where we put sponges on long ropes but left them out in the marine environment. And in those cases, we managed to maintain the same level of production of the compound we were interested in that study. So it's on a case-by-case basis, and there's a lot of work that needs to be done on that as well.
Thank you, Prof. The questions in the chat room is already answered by you. But I welcome from audience. If you want to say something to Prof. Russell, it's a good opportunity for you. And Prof. Russell welcome more questions actually. However, there is now 10 p.m. means late night for Prof. Russell. Yeah, I wait for maybe 3 minutes to allow you raise hand or share question if you wish.
Ibu.
Who is it? Yeah, Bu Weni.
Tanya.
Miss Weni, please.
Ibu, maaf kalau ini pertanyaan awam karena kita biasa di klinis. Mohon maaf pakai bahasa Indonesia ya, Ibu.
Ya, nanti ditranslate.
Untuk Prof. Russell, tadi dikatakan spong jenis spong yang Ehrlichiana, contohnya Ehrlichiana strobilina, itu kan akan menghasilkan mikroba, contohnya Actinomycetes. Nah, apakah setiap jenis ini menghasilkan Actinomycetes dan apakah setiap wilayah itu akan sama? Contohnya, kalau ditemukan di wilayah lautan Jakarta dengan lautan Makassar, apakah kandungannya itu sama? Itu, Ibu. Terima kasih, Ibu.
I will try to translate, Prof. So, Miss Weni wants to ask you, you said that sponge is a source of actinomycetes. Will actinomycetes from different environment will have the same or potent activity? Probably that's what I catch.
Yes, I've done my first work on marine actinomycetes was not really marine, it was estuarine. It was isolating actinomycetes from the sediments of the Chesapeake Bay, which is a very big estuary on the east coast of the United States, and Baltimore is on the Chesapeake Bay. And we got a lot of interesting actinomycetes from marine sediments as well. I think there are many different sources, but for some reason, sponges have been really good in yielding novel actinomycetes. And because sponges have so much interesting chemistry and they have so many bacteria in them, I think your chances of finding bacteria that are making novel compounds may be a little bit higher in sponges. But I would encourage you, if you, any of the audience, have other organisms, marine invertebrates that they are very interested in and that they know they're knowledgeable about them already, it's certainly worth thinking of doing some microbiology and natural products discovery on your favorite marine invertebrate, because there will be things to be found everywhere. We already heard about the promising work from the holothurians, the sea cucumber. So there are lots of possibilities.
Prof, probably what Miss Weni wants to know is whether actinomycetes from marine environment and from the land, for example, will have the same potent bioactive or not, like that.
So that's not really been found. We are hesitating because I've also done some work on fungi when I was doing research in Australia. In the case of fungi, we discovered quite a few fungi. We isolated them from marine organisms and then found they were very similar to terrestrial organisms and they made similar compounds. We have not really found that with actinomycetes. We find a lot of novel actinomycetes from marine sources that have not been isolated from the terrestrial environment.
Oh, okay. Okay, so Miss Weni, I think you have got the complete answer. So novel is from marine, but from land maybe not novel. Sorry, make it easier to understand. All right, so everyone.
Terima kasih, Bu Norma.
Yes, you're welcome. Now it's 10:11, Prof. So I think we are reaching the end of this general lecture. I would like Dr. Sri Darmawati. I hope Dr. Sri Darmawati is still here.
Okay.
Yeah, to give
Ada, Bu. Yeah.
closing statement.
Okay. Thank you for your time for me. Prof. Russell, Honorable, I would like to present my gratitude, especially from Prof. Russell, and second to Prof. Oki Rajasa. Thank you for being here with us, Prof. Russell, despite your busy schedule as Director of Marine and Environmental Technology Institute. And I hope this event will not be once and the last, but will be followed by other great events. I hope we could continue our cooperation through academic exchange, through visiting scientists, postdoc, and research activities. Also to Prof. Oki, pada Prof. Oki dan semua audien dari mahasiswa S2 Ilmu Laboratorium Klinik beserta audien yang lain yang telah gabung di Zoom ini. Terima kasih atas atensinya, atas waktunya untuk bergabung, and I hope we could meet again in greater occasion in near future. Thank you, Prof. Russell, Prof. Oki, and the other. Bu Dewi, terima kasih waktunya sudah mengkonektorkan kegiatan ini. Ya, dan terima kasih juga untuk semuanya. Semoga kegiatan ini tidak hanya berakhir di sini, dan tentu kami mohon Prof. Oki juga bisa membuka peluang bagi kita semua untuk bisa memanfaatkan punya negara yang Prof. Oki sekarang sedang kelola. Ya. Dulu di Kemendikbud, sekarang di LIPI meskipun gabung di satu BRIN. Ya, tentu saya yakin Prof. Oki terbuka untuk kita semua. Demikian pula Bu Dewi. Thank you for all. Assalamualaikum warahmatullahi wabarakatuh.
Thank you. Thank you very much. It was a great pleasure for me to see my old friend Oki again after many years, and I've enjoyed this very much. Thank you to all of you for making this opportunity for me and for all your attention. It was wonderful to see such a big audience and have such great questions. I really enjoyed it. Thank you.
Thank you.
Thank you, Russell. We will be in touch. Thank you very much for the opportunity.
Thank you.
Stay safe and healthy.
You too, all of you. Yeah.
Okay.
Good night.
Salam sehat semuanya.
Terima kasih. Ya, Assalamualaikum.
Terus an email for certificates Prof. Oki and Prof. Russell. Yeah.
Thank you.
Bye-bye.
Bye.
Bye. Makasih ya Bu Dewi.
Sama-sama Bu Cici.
Ya. Terima kasih juga Bu Norma udah
Bu
memandu
Cici
acara
hebat,
ini.
Bu Norma hebat.
Ini saya di Bali Bu Dewi.
Yang di Bali.
Ya, saya sengaja hari ini off kegiatan keluar. Saya di hotel ini kan ada kegiatan ini.
Untung acaranya sukses ya Bu Cici, jadi nggak ini ya.
Ya, nggak rugi. Nggak rugi.
Iya.
Oh, Bu Ciki juga gabung ini ya.
Selamat Bu Cici, Bu
Makasih Bu
Norma.
Ciki.
Bu
Ya,
Dewi,
yo,
terima
makasih.
kasih.
Ya,
Izin
Mas Ruli,
wonderful.
makasih Mas Ruli.
Izin live ya Bu Cici.
Ah, iya ya.
Ketemu ya Bu
Udah
Cici.
ada presensinya ya Bu Norma.
Ada.
Kembali nggih untuk Bu Ciki.
Ada,
Iya,
mungkin bisa
nggih.
di-share di Bu Ceki.
Khusus untuk Bu Ciki nggih. Ini
Ya,
thank
nanti
you very
biar
much.
di-share, nggih.
Oh
Ya,
iya,
Bapak
oke.
semua atas kehadirannya. Nggih, nanti
Oke, makasih nih Pak Muhammad Hendri, siapa dari mana ini?
ya ada Bu dari LIPI, ada dari mana ini asosiasi.
Ya, terima kasih udah
Sama-sama
gabung.
Ibu, dari Palembang Ibu.
Oke,
Luar
dari Palembang,
biasa.
dari Unsri.
Siap. Itu
Oke.
ada narsumnya itu co-promoter saya tadi, Prof. Oki.
Oh gitu.
Iya.
Oh iya.
Oh, di
makanya
biologi
diminta
ya?
ngikutin.
Di ini Ibu di kelautan. ilmu
Oh, di kelautan.
Di
Unsri, saya punya temen Mbak Hayis Nita. Di
Mbak siapa?
Unsri. Hayis Hari Wijayanti.
Oh iya, ketua ini sekarang beliau, ketua program S3.
Oh
Udah
ya,
Penari.
oh,
Dari
teman
UNAIR kalau nggak salah.
seangkatan. Teman seangkatan.
Siap,
Salam
siap,
ya.
Siap Bu, nanti saya sampaikan salamnya.
Oke, makasih udah
Sama-sama Ibu.
gabung. Makasih ya Bu ya.
Yes,
Selamat
sukses
ya.
yes, sukses.
Amin.
Ini saya
Makasih.
ngisi apa? Ngisi
Oh,
yang dikirim Bunda
Ya,
Norma.
oke. Makasih. Jen live ya.
Sukses, jaga kesehatan ya Bun.
Ya.
Iya Bu, saya end ya Bu. Iya, boleh di end. Saya kira
Kira di end aja. Oh Mbak Fitri ya?
Mbak Fitri, ini ada link YouTube. Nanti kalau mencari record acara ini ada di link YouTube tersebut, tapi di hari Senin.
Ada, saya juga ada anu kok. Nganu.
Iya, karena hari libur Bunda.
Oke.
Baik, kita akhiri, kita tutup ya Mbak ya. Ya. Monggo Bu Ciki.
Eh.
Halo. Makasih.
Bun, tak sih Bu Norma tak foto dulu. Satu.
Ya, Bu. Oh.
Ya, spesial. 1, 2. Cantik. Udah, deh, tak kirim Bu Norma, udah saya itu.
Alah, ya Allah.
Ini luar biasa.
Belum ditutup. Begitu kita